So vaccines are a little different, and there is a current study that's recently been closed now because it accrued all their patients in almost in kind of record time. And it's mRNA vaccine technology that was developed for COVID that's actually being used in cancer therapy right now. And so it's how the mRNA vaccines work, and it's being tested right now in melanoma, for example. The the vaccine is actually a personalized vaccine. It's based on the identification of a very important neoadjuvant that may be present in the tumor, and that protein or that actual protein is being actually decoded, and you can make that protein using mRNA. So it's just like the mRNA vaccine for COVID. It makes the body it helps the body make this protein so it can be identified as foreign, so the body can make antibodies against not only antibodies, but also probably killer T cells against those proteins. And this is being done in melanoma, for example, and likely going to happen in other solid tumor types as well. And so this platform using mRNA vaccine, more than almost a 2,000 patient study is now being completed, where patients received the vaccine in the postoperative adjuvant setting after receiving immunotherapy up front. So the study was that the patient received immunotherapy up front, they had surgery, and then they continued on the immunotherapy afterwards, and then they added the vaccine. So that's different than oncolytic immunotherapy. Its mechanism to improve the immunologic response is completely different than the oncolytic viruses, which are injected directly into the tumor. Our group has participated in that vaccine trial. We've done several of our own institutional neoadjuvant trials looking at different immunotherapeutic combinations. Like one of the combinations that I told you was this ipilimumab with anti-PD-1, where there's another checkpoint blocking agent called LAG3 that is important as well, and it has less side effects than the anti-CTLA4. So the combination of anti-PD-1 plus anti-LAG3 is another immunotherapeutic combination that's been studied in the neoadjuvant setting. So we also have studies where we're looking at some of these correlates for complete response, for example, circulating tumor DNA, for example. So many of our neoadjuvant trials have these biologic correlates, that's called translational research, where we're studying the level of circulating tumor DNA as a potential marker for either lack of response or a complete response. So there's several avenues in which research is ongoing to try to better understand who's going to respond and who's not going to respond, and how to augment that response if they're going to respond, and better ways to determine how much systemic therapy the patients actually need. So less side effects, less financial toxicity. So these are several parallel endeavors that are going on at the same time. And where everyone is now using this neoadjuvant platform in which to do that. And I mean, the advantage for these stage 3 patients is that we have such easy access to the tumor, because the stage 3 patients are the ones that have kind of bulky lymph node metastases, and these are easily accessible to do like biopsies before and during therapy. And it allows us to have a much easier ability to identify these important biomarkers.